Hey everyone, Raman here. Today I want to give you a quick review of a rejuvenation technology from an American startup called Mitrix Bio. You already might have seen the news about their mitochondrial rejuvenation therapy and their promising first patient. But today I want to focus on the technology itself. As you probably know, mitochondria are often called the powerhouses of our cells. They produce ATP, the main energy molecule in our body. But here's the thing, it turns out that besides generating energy, these mitochondria are incredibly busy. They help produce hormones, build fatty acids, and even play a role in our immune system. Inside them, key processes happen. The metabolism of fatty acids, biosynthesis of heme, which is essential for hemoglobin, and the assembly of iron-sulfur clusters. Now, don't let that complex term scare you. These clusters are just vital cofactors. Think of them as tiny tools that are absolutely essential for enzymes to work properly, especially those involving in DNA replication and our cellular respiratory chain. They are like dispatchers in a crisis. If they see that energy is critically low, they can issue a command for cells to self-destruct. And here's the most fascinating part. Mitochondria have their own DNA, and this DNA is passed down only through a maternal line, so you inherit your energy power directly from your mother. Here's another cool fact. Mitochondria are direct descendants of alpha protobacteria that entered into symbiotic relationship with eukaryotic cells over 1 billion years ago. Essentially, one organism swallowed another organism but couldn't digest it, so they started to live together. And that, according to scientists, is how mitochondria came to be. Also, this is why mitochondria in all living beings on the planet had the same structure. This origin also explains why they have their own circular DNA, so-called mDNA. And this is where the main problem lies. If errors like mutation occur in this unique DNA, the serious breakdown begins. In children, this can lead to a severe hereditary disease where the body literally cannot produce energy and develop properly. In adults, malfunction in these batteries are linked to a whole host of problems, from diabetes and depressions to Parkinson's and Alzheimer disease. And the saddest part is that as we age, the number of faulty mitochondria keeps growing. When we're young, our body knows how to sort them out and recycle them like garbage. But later, this cleanup system breaks down. Imagine it, an old leaky power station not only works poorly, but also dumps toxic waste into the environment. This waste damages other power stations, and this all spirals into a vicious cycle of aging. Some scientists believe that this vicious cycle is one of the main reasons why we age and develop chronic health issues. Hypertension, stroke, diabetes, all of these can be a consequence of our internal batteries running low and starting to leak toxins. Everything I've just described is a part of free radical theory of aging. So, what we can do about it? Science has a bold idea. What if we just transplant the new mitochondria? And that's exactly the core idea behind Mitrix Bio's technology. They propose growing mitochondria in a special bioreactors, multiplying them, and to put it bluntly, just inject them into the bloodstream. It's essentially like replacing an old, worn-out battery in your phone with a new, powerful one. Sounds like science fiction, but experiments like this have been going on since 80s. Theoretically, this could grant us healthy longevity and free us from most age-related diseases. And on the Mitrix Bio website, there's a really interesting section. Researchers have discovered in the last decade that mitochondria don't just sit inside the cell. They are actually constantly being transported throughout the body. Every day, hundreds of billions of them are transported via the bloodstream to the brain, heart, and other organs to supplement cells in need. Many of these mobile mitochondria are encapsulated in extracellular vesicles, what they've called mitlets. These mitlets are like the body's natural fountain of youth, used to balance and maintain healthy cellular energy, and it's incredible evolutionary adaptation to increase the survival and longevity of species. So that's the gestator technology. In my view, this is incredibly promising, and I have a personal feeling that they are on the right track, but it's still a fairly distant future. However, knowing that Synthesit had a significant impact on mitochondria, I've decided to dig into the reasons behind these age-related changes in mitochondria, and I discovered something very interesting. Turns out that the disposal of old mitochondria is regulated by a system called mitophagy, from the words mitochondria and phagy, meaning to eat. I mentioned this before, but if we're looking for main culprits, there are two key players, a protein PINK1 and parkin. They are in charge of mitophagy. So what breaks down with age? The problem isn't that PINK1 and parkin genes themselves mutate. The main age-related issue is that the efficiency of the entire mitophagy system decline. The first is reduced activity. The production and activity of the PINK and parkin proteins can decrease with age. The alarm signal gets quieter and the markers work slower. And the second is traffic jams. Even if the damaged mitochondria are targeted, the delivery system to the lysosomes can fail. Imagine the garbage truck getting old and making fewer trips. The third reason, too much garbage. With age, the number of damaged mitochondria becomes so large that the system simply can't handle the workload. It gets overwhelmed like a cleanup crew after a massive party. As a result, instead of being recycled, these decrepit mitochondria keep hanging around in the cells like bad neighbors. Not only do they produce energy poor, but they also constantly damaging neighboring still healthy mitochondria. And this is exactly that vicious cycle of aging. Mitochondria getting more damaged, this leads to overloading the mitophagy system, and this leads to even more damage. And here's what's interesting. In our research at MicroLife Biotech, we didn't measure the expression of these specific genes, though now we understand we probably should have. However, there are several powerful indirect mechanisms for which Synthesit can positively affect PINK-parkin system and mitophagy as a whole. Here are the key findings from the report that points to this. The first mechanism is the reduction of oxidative stress, because it's the main trigger for healthy mitophagy. The PINK-parkin system activates in the response to mitochondrial stress, often caused by excess of ROS, reactive oxygen species. If the stress is chronic and excessive, the system breaks down. One of the strongest effects of Synthesit is powerful activation of antioxidant pathways. We observed a sharp increase in the expression of genes like NRF2, the master regulator of antioxidant response, SOD2, superoxide dismutase, which neutralize superoxide radicals right inside the mitochondria, HMOX, a critically important enzyme for stress protection, GPX1, CAT, TXN, and many others. As a result, by suppressing excessive reactive oxygen species production, Synthesit prevents chronic overload and damage to the PINK-parkin system. This creates conditions where the system can work correctly, activating when it's truly needed, instead of being a constant state of emergency alarm. The second is improving overall mitochondrial health. Healthy mitochondria gets damaged less often and trigger PINK-parkin cycle less. The report shows that Synthesit significantly improved mitochondrial function, which allies with Zhovt-Radek's research on rats. Specifically, enhanced biogenesis, increased expression of genes related to creating new mitochondria like PGC1 and NRF1, increased efficiency, higher levels of CPT 1A point to more efficient energy production with fewer harmful byproducts like ROS. Also, there's an indirect impact, the strengthening the overall mitochondrial population. Synthesit reduces the total workload on the quality control system. The healthier the mitochondria are, the less garbage needs to be disposed via PINK1 and parking, this preventing system from overload. And the third mechanism is the shift to anti-inflammatory state, so-called M2 phenotype. The activation of M2 macrophages, as shown in the report, is directly linked to improved metabolism and mitochondrial health. In contrast, the pro-inflammatory M1 phenotype is associated with impaired mitophagy and the accumulation of damaged organelles. To summarize the key takeaways, it reduces the background load thanks to a powerful antioxidant protection. It improves the overall conditions of mitochondria, which reduces the number of breakdowns. It creates metabolically healthy cellular environment, so-called M2 phenotype, that is favorable for mitophagy. In essence, Synthesit doesn't target the symptoms by trying to directly stimulate PINK parking system, but rather deal with the cause. It tackles the main factors like oxidative stress, poor mitochondrial health, and others that leads to a failure in the system. This is more fundamental and likely more effective approach to maintaining long-term cellular quality. So now we have a clean goal to specifically look at Synthesit impact on PINK parking system. It's for the full confirmation of this hypothesis. Although the results seems obvious, the scale of the changes still matters. Thank you very much.